Hello, my dear students. Welcome you all to Sri Jayendra Rank Zone classes. My dear students, today we are going to start with human heart. But before talking to that topic, we will be discussing the last part which was left over in the previous video, that was the coagulation cascade. The blood coagulation, scientifically known as hemostasis, is followed by two types of pathway. One is the intrinsic pathway and the extrinsic pathway. There are 13 clotting factors. We discussed all the clotting factors in the previous lecture. The intrinsic pathway and the extrinsic pathway, they are activated by different types of stimuli. Different stimuli activates both the pathways. Okay. There are some common factors which are required, especially the common pathway begins from the factor number 10th. So I would like to just revisit all the types of factors once again. Factor number 1, fibrinogen. Only the names I will tell you. Fibrinogen, which is required in both extrinsic and intrinsic pathway. Factor second, prothrombin, required in both the pathways, extrinsic and intrinsic. Factor number 3, thromboplastin or tissue factor, required in both the pathways. And this pathway, factor number 3, disintegrates that which type of cascade, which type of pathway will be followed, whether intrinsic or extrinsic. As if the platelets release thromboplastin, then it is intrinsic pathway. If the endothelial wall, means the blood capillary wall releases, blood vessel wall releases the tissue factor or the thromboplastin, then it is extrinsic pathway. Then factor number 4, calcium. Factor number 5, proacylidine or stable factor, labile factor. Factor number 7, 6 is unknown. Factor number 7, proconvertin or stable factor. Factor number 8, anti-hemophilic factor. The deficiency of anti-hemophilic factor, factor number 8, causes hemophilia A. Then factor number 9, Christmas factor. Okay. Then factor number 10, stored power factor. Factor number 11, plasma thromboplastin antecedent. Or factor number 12, Hegman factor. Factor number 13, fibrin stabilizing factor. And other two accessory factors, precalciferin and kynogen. Okay, precalciferin and kynogen. Now, coming to the pathway again, just a minute. Yeah. So extrinsic pathway, it activates with factor number 3, okay, converts to factor number 7, and factor number 7 requires calcium, okay, to be converted into factor number 10, okay. Then factor number 7 also activates, in the presence of calcium, also activates factor number 9. Collagen helps to activate factor number 12. Factor number 12 converts to factor number 11. Factor number 11 then converts to 9. 9 to 10. Factor number 8 and calcium along with factor number 9 required for the conversion to factor number 10. I repeat, factor number 8 plus calcium plus 9 converts to factor number 10. Factor number 5 and calcium, they are required for the activation of factor number 10 to prothrombin. Factor number 10 in the presence of factor number 5 and calcium. Okay, to prothrombin, prothrombin converts to thrombin. Okay, prothrombin converts to thrombin. Thrombin converts fibrinogen into fibrin. Very important. Okay, now thrombin, it converts platelets to form a platelet plug, that is clot, and it helps in the stabilization of cross-linked polymers of fibrin. Okay, cross-linked polymers of fibrin along with fibrin as well. So the red arrows indicates the clotting factors needed to be present in order to activate the next part of the clotting cascade. Factor number 13, fibrin stabilizing factor is required for the conversion of fibrin degradable polymers into fibrin cross-linked polymers. So these are fibrin degradable polymers which are soluble to fibrin cross-linked polymers. Fibrin cross-linked polymers. Okay, you have to remember the name of the factors and the function and the requirement, whether intrinsic or extrinsic. Please remember about factor number 10. The common pathway starts from factor number 10. Factor number 7 also converts to 10. 7 converts to 9 as well in the presence of calcium. And factor number 9, 8 and calcium converts into factor number 10. Okay. So this is all about neither, I mean, there is no requirement of going into deep of these cascade. Okay. So one thing I just want to tell you that factor number 8 deficiency causes hemophilia A, factor number 9 deficiency causes hemophilia B, and factor number 11, which is responsible for plasma thromboplastin antecedent or anti-hemophilic factor C. Anti-hemophilic factor C, the deficiency causes anti-hemophilic factor C or when will brand disease. When will brand disease. Okay, understood? Now, functions of blood. Blood helps to transport the food materials. Blood transports the digested food from the alimentary canal to different body cells. It helps to transport food from alimentary canal to different body cells. Transport of respiratory gases. Oxygen is carried from the respiratory organs to the tissues, and carbon dioxide is carried from the tissues to the respiratory organ by blood. Transport of hormones. Hormones are carried by blood from the endocrine glands to the place of use, means the target organs. Target organs. Transport of excretory substances. Blood transport to carry excretory matter to the kidney or the excretory organs for their selective reabsorption and secretion. For their selective reabsorption and secretion. Transport of heat. Blood allows the transfer of heat from the deeper tissue to the surface of the body where it can be lost or dissipated. Heat dissipation to maintain the body temperature. To maintain body temperature. Defense against infection. White blood corpuscles or leukocytes are phagocytic in action. However, certain blood corpuscles produce antitoxins to neutralize the toxins released by foreign germs. Okay, certain blood corpuscles produce antitoxins to neutralize the toxins released by foreign germs. Temperature regulation. Blood maintains the body temperature to a constant level after distribution of heat within the body. Water balance. Blood maintains water to a constant level by bringing about constant exchange of water between circulating blood and the tissue fluid. So tissue fluid and circulating blood, they help to exchange water contents to maintain the overall water in the body. Maintenance of pH. Blood helps to regulate the pH by the buffering action of bicarbonate ions. Prevention of excessive loss of blood. When any part of the body is injured, loss of blood is prevented by the formation of clot. When any part of the body is injured, loss of blood is prevented by the formation of clot. Blood clotting cascade, I have already explained. Helps in healing. Blood maintains necessary supplies for the repair of damaged tissue. Okay, eosinophils and basophils help in the healing of wound. Very important. Eosinophils and basophils, they help in healing of wound as they are antimicrobial, they are anti-inflammatory. Okay. Maintenance of physiological cooperation. Blood maintains a physiological cooperation between the parts of the body by circulating from one to the other parts. They help the different body organs to be in a synchronized manner. So, to maintain the water, to maintain the pH, prevention of excessive loss of blood, helps in healing of wound. Eosinophils and basophils, they help in healing of wound. They maintain the physiological cooperation between different organs to maintain a synchronization between the working of different organs. Blood groups. Carl Landsteiner reported first time ABO blood group system in human being in 1900. AB blood group was found out by D. Castellan and Steny in 1902. Okay, so Carl Landsteiner reported first time ABO blood group in human beings 1900. AB blood group was found out by D. Castellan and Steny in 1902. If the blood transfusion is made between an incompatible donor and the recipient, reaction of antigen on the cells and the antibodies in the plasma produce clot that clog capillaries, that block the capillaries. So basically, the surface of RBC is having antigen or agglutinogen, and the plasma is having antibody agglutinin. Okay, so these agglutinogen and agglutinin, they combine with each other to make a clot if there is any mismatch in blood transfusion. If the blood group is A, if the antigen present on the RBC is A, then blood group A. If the antigen on RBC is B, then blood group B. If the antigen is AB, then the blood group is AB. If the antigen is neither A, neither B, neither AB, then it is O. Okay. Genotype for A is IA, where I represents the agglutinogen. Okay. So IA or IAIO. IAIA is a homozygous condition. Homozygous. And IAIO, heterozygous. Both represents A blood group as O comes to be recessive in terms of A, B and AB. B blood group, same IB or IBIO. AB blood group, only one condition, IAB. 50% of RBCs are having A blood group, 50% of RBCs are having B blood group. So IAIB represents codominance, as 50% of RBCs are having A blood group and 50% are having B blood group. Okay. IO, only one condition, homozygous recessive. Homozygous recessive. Antigens in RBC, IA, B, AB, none. So O blood group is not having any antigen. B blood group is having only B, A is having only A, and AB is having both. Receive blood from A blood group A, person having a blood group A can receive the blood from both A and O. O is a universal donor. B and O. AB can receive from O, A, B, and AB. It is a universal recipient. And O can receive from only O. Blood group A can donate blood to A and AB. B can donate to B and AB. AB can donate only to AB. Okay. And O can donate to anyone because O is not having any antibodies. O is not having any antigen. Okay. Blood group AB is not having any antibody. Blood group B is not having any antigen. Okay. Percentage in humans, 41% of A blood group, 10% of B blood group, 4% of AB blood group, and 45% of O blood group. Okay. Rhesus blood group or RH factor. A protein named rhesus antigen is present on the surface of red blood corpuscles in many persons. It was discovered by 1940 by Landsteiner and Wenner in the blood of rhesus monkey. Hence its name. 85% of humans, mean more than 93% Indians have blood protein called Rh factor, Rh plus. Other without the factor is called Rh negative. The ones which are having Rh antigen, if Rh antigen is present, then Rh positive. If not, then Rh negative. Okay. Rh is tested with the help of Rh antiserum or plasma containing Rh antibodies. Rh is tested with the help of Rh antiserum or plasma containing Rh antibodies. Agglutination occurs in Rh positive cases while non-agglutination shows Rh negative nature. Agglutination happens when Rh antigen is present, means Rh positive cases. Non-agglutination shows Rh negative nature. Rh plus is dominant over Rh negative. The antigen formation is determined by a dominant allele capital R. It receives rise, it gives rise to Rh plus condition. Presence of double recessive small r does not form antigen so that the individual is Rh negative. If the capital R is present, okay, then Rh positive condition. And if the double recessive small r is present, then no antigen is present, means Rh negative. Rh positive blood is given to Rh negative person, produces anti-Rh factor A. The first baby is saved due to the late development of anti-Rh factor A. Okay. Now I will tell you the Rh incompatibility in especially in the pregnant ladies, okay, having a Rh positive baby, and that was the first delivery. And what about the second delivery? Okay. The second Rh positive baby will either die in the fetus stage or born anemic with several abnormalities due to disintegration of red blood cells, erythroblastosis fetalis, by anti-Rh factor small a. Anti-Rh globulin is available to overcome that effect and subsequent and consequent production of excess bilirubin. Anti-Rh factor, okay, anti-Rh globulin is available to overcome that effect and consequent production of excess bilirubin. The latter can damage the brain of the infant. Excessive amount of bilirubin can damage the brain of the infant, however, can damage the brain of the infant. However, the reverse does not have the effect. Oswald Hope Robertson is the creator of the first blood bank. Now, what is Rh incompatibility? If a Rh positive male marries to Rh negative female and the baby is Rh positive, then the baby develops antibodies against Rh negative. Okay, or we can say the mother develops antibodies against Rh positive. Okay, always remember the Rh positive is antigen, so it cannot develop antibodies against itself. So it is the mother or the maternal blood, okay, just beside the placenta, who develops the antibodies against the Rh positive fetus. The first delivery is safe. Why? Because till the first delivery there is no disruption in the umbilical cord. There is no disruption in the junction of maternal and the placental villi or the tissues. But the moment after the first delivery happens, the umbilical cord is cut. There is a internal damage to internal damage which basically uh, connects the maternal blood to the fetal blood. So, making of the antibodies inside the mother will travel through those pores to the fetal blood and can damage it. Okay, that is why the second baby comes to be abnormal, or shows different types of fetal abnormalities known as erythroblastosis fetalis. Nowadays, anti-Rh globulin or D vaccine or anti-D factor is used to overcome that effect. Okay, excessive amount of bilirubin can damage the brain of the infant, and a very important radiographic feature is shown by these babies suffering from erythroblastosis fetalis, that is Rh hump. Rh hump. RH hump is basically a kind of hematoma or blood-filled overgrowth seen in the gingival region and in the mucosal region, mouth. Okay. Importance of blood groups. Knowledge of the blood group is essential for blood transfusion. For the correct blood transfusion, it should be there that person should know about the blood groups. Okay. RH compatibility is required for both marriage and transfusion in order to prevent erythroplastosis. Preliminary information about disputed parentage and progeny is provided by blood grouping. Okay, blood groups is used for forensic identification of blood stains. Okay, what is the importance of blood groups? Knowledge of blood groups is essential for blood transfusion. RH compatibility is required for both marriage and the transfusion in order to prevent erythroblastoma fetalis. Preliminary information about disputed parentage and progeny is provided by blood grouping. Blood grouping is used for forensic identifications of blood stains. So that is all about the blood groups. And now moving to the next slide, now we will discuss human heart, anatomy of human heart. Okay. It is a reddish conical muscular mesodermal in origin. Hollow organ about 12 centimeter in length, 9 centimeter in breadth, weighs about 300 grams and lies behind the sternum in the mediastinum space of thoracic cavity, in between two lungs. Broader base is upward. Okay. So it is a reddish conical muscular mesodermal hollow organ. It is mesodermal, it is muscular, it is reddish, it is conical, about 12 centimeter in length, 9 centimeter in breadth, and weighs about 300 grams and lies behind the sternum in the mediastinal space of thoracic cavity in between two lungs. Broader base is upward. The base is broad and it is upwards. Okay. The mammalian heart comprises of four complete chambers, two ventricles, and two auricles. Auricles are known as what? Atria. Heart wall consists of connective tissue. Heart wall basically consists of connective tissue, blood vessels, and cardiac muscle fibers. Heart wall mainly consists of connective tissue, blood vessels, and cardiac muscle fibers. The latter form a cross-connected network of smooth passage of constriction wave. The cardiac muscle or myocardium does not tire due to alternate rest and activity, non-formation of lactic acid. There is no fatigue which is found in myocardial walls because of alternate rest and activity, okay, and non-formation of lactic acid because of high amount of myoglobins and mitochondria. So heart wall consists of connective tissue, blood vessels, cardiac muscle fibers. The latter form a cross-connected network of smooth passage of constriction wave. The cardiac muscle of myocardium does not get tired due to alternate rest and activity and non-formation of lactic acid. The mammalian heart comprises of four complete chambers, two ventricles, and two auricles. Heart is covered by double fibrinous sac or pericardium. It is covered by pericardium. The two components of outer non-distensible tough fibrous pericardium prevents excessive expansion of heart, and inner thin serous pericardium. Outer is non-distensible tough fibrous pericardium. So outer is fibrous, tough and non-distensible, and inner is serous. Okay. Serous pericardium has two thin secretory membranes, outer parietal pericardium and inner visceral pericardium. Serous is further divided into outer is parietal and inner is visceral. It encloses a narrow pericardial activity having pericardial fluid for frictionless movement, protection from shock and mechanical injury. Serous pericardium has two thin secretory membranes, outer parietal and inner visceral. It encloses a narrow pericardial cavity called pericardial fluid, which provides frictionless movement of the heart walls, protection from the shock and mechanical injury. There is depression or coronary sulcus between atria and ventricles. Atria and ventricles, they have a small sulci, okay, or a depression known as coronary sulcus. Interatrial sulcus two parts, anterior and posterior, between two ventricles. Okay, so there is a depression or coronary sulcus between atria and ventricles, interatrial sulcus and interventricle sulcus. Okay, so interatrial sulcus, it basically divides two parts, that is anteriorly and posteriorly between two ventricles. Coronary arteries are housed in these sulci. Coronary arteries are present in these sulci, in these interatrial and interventricular sulci. Okay, or coronary sulcus. Coronary sulcus is present between atria and ventricles, and interatrial sulcus. Okay, they supply blood to walls of the heart. Okay, coronary arteries, they provide blood to the walls of heart. Atrial appendages is protruded part of atria which overhangs the ventricle. Low ridges occur internally in the region of atrial appendage. They are called musculi pectinati or pectini muscles. Atrial appendages mean part of the atria which overhangs the ventricles. Low ridges occur internally in the region of atrial appendages. These are called musculi pectinati. Blood vessels connected to heart are known as great blood vessels. Blood vessels connected to the heart are known as what? Great blood vessels. Okay. Deoxygenated blood flows through the right half of the heart, mean the right part, and oxygenated blood to the left part of the heart. The right and the left atria are separated by interatrial septum. It bears a depression called fossa ovalis in the area of fetal opening called foramen ovale. So foramen ovale is a opening in the fetal life which gets obliterated, which gets closed to form fossa ovalis. It bears a depression called fossa ovalis. So deoxygenated blood flows on the right side, oxygenated blood flows on the left side of the heart. The right and the left atria are separated by interatrial septum. It bears a depression called fossa ovalis in the area of fetal opening called foramen ovale. Right atrium or auricle receives deoxygenated blood from the superior vena cava, upper part of the body, inferior vena cava, middle and lower part of the body, and coronary sinus, heart walls. Okay, the basal valve occurs at the opening of coronary sulcus, okay, and eustachian valve at the opening of inferior vena cava. The basis valve occurs at the opening of coronary sinus. At the opening of coronary sinus, there is a presence of, uh, valve of basis, and the eustachian valve at the opening of inferior vena cava. Backflow in the superior vena cava is prevented by obliquity of opening. The left atrium auricle receives oxygenated blood from the two lungs through four pulmonary veins. Right and left ventricles are separated by interventricular septum. So the right atrium or right auricle receives deoxygenated blood from the superior vena cava, upper part of the body, inferior vena cava, middle and lower part of the body, and coronary sinus, means the heart walls. The basis valve is basically at the opening of coronary sinus, and eustachian valve at the opening of inferior vena cava. Okay. Mm, Thebasian valve, I think it's Thebasian valve. It is Thebasian valve. Thebasian valve guards superior vena cava. Superior vena cava. Okay. So backflow is in superior vena cava is prevented by obliquity of opening. The left atrium auricle receives oxygenated blood from two lungs through four pulmonary veins. The right and left ventricles separated by interventricular septum. Left ventricle is larger because of physiological hypertrophy, includes the apex part, and has extra thick wall as compared to the right ventricle due to its mechanical requirement of pumping oxygenated blood. As left ventricle, it is having major workload to pump the blood to all the parts of the body, and that is why physiologic hypertrophy happens. Physiology, physiologic hypertrophy is present in left ventricle. Please rectify this term. This is Thebasian valve which guards the coronary sinus, which guards the coronary sinus and these inferior vena cava. Okay, the Thebasian valve in the coronary sinus and this superior vena cava region. Okay, now 12 parts of the body. Walls of the ventricle possesses the network of low ridges or columnal carne, and few larger muscular projections or papillary muscles or musculi papillars. Okay, so the left ventricle basically is larger. It includes the apex part and has the extra thick wall as compared to right ventricle due to its mechanical requirement of pumping oxygenated blood to all parts of the body. Walls of the ventricle possesses a network of low ridges or columnal carne Okay, and a few large muscular projections called papillary muscles or musculi papillaris. Okay, right ventricle contains a moderate band that extends between upper papillary muscle and interventricular septum. Atria opens into ventricles through atrioventricular apertures is guarded by the valves. Mitral valve and means the bicuspid and the tricuspid valve. Right atrioventricular aperture is guarded by tricuspid valve possessing three flaps, and left atrioventricular aperture is guarded by bicuspid valve or the mitral valve possessing two flaps. Okay. The flaps of the valves are held in their position by fine elastic cords or chorda tendini connected to the papillary muscles. Okay. The chorda tendineae basically connects the interventricular, these atrioventricular flaps, okay, to papillary muscles. Left ventricle opens to aorta. The opening is guarded by semilunar valves, aortic semilunar valves between two. Okay, this is the human heart. You can see uh, this is the esophagus at the back. This is the trachea, and these are the three parts of the great blood vessel of the aorta. That is brachiocephalic artery, left subclavian artery, and left common carotid artery. So left common carotid. So aorta is having three branches mainly: brachiocephalic, left subclavian, and left common carotid. Okay. Left pulmonary arteries, four left pulmonary veins, left atrium, bronchi. This is bronchi, semilunar walls between pulmonary artery and aorta. Atrioventricular wall, mitral valve on the left side. Okay. This is descending aorta, esophagus, inferior vena cava, guarded by eustachian valve. Superior vena cava, right pulmonary arteries, right pulmonary veins, right atrium. Okay. Uh, Superior vena cava and coronary sinus, they are guarded by Thebesian valve. Thebesian valve of basal valve. Okay, so this is the overview of the picture. Okay, the blood comes to the right atrium to right ventricle, then to lungs, then again from pulmonary vein to left atrium to left ventricle, and then aorta. Functioning and physiological part will be discussed in the next lecture, my dear students. Till then, please take care. Bye bye. Stay safe. Stay healthy.